From Microbe TV, this is Immune, episode number 17, recorded on February 12th, 2019. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back, listeners.
Did you get any skiing in yet?
We did. I've been skiing almost every weekend. Yay.
Nice. Where do you go, to Greek Peak?
We do, yeah. The kids are getting much better.
Cool.
Yeah, they have black diamonds with jumps and grinds and things, and I'm watching my children go off and fly 10 feet in the air. It's a little freaky, but they're having a great time.
Also joining us from the ski capital of the world, Durham, North Carolina, Steph Langel.
Hey there. Yes, skiing may be water skiing if it was warm enough, but I'm definitely, we were talking earlier before the podcast started, I've become the person that nobody really wants to hear the weather from. I think last week it got up to like 75. So right now it's a little cold. It's a little cold, and rainy, but it's not too bad.
Today is Darwin Day.
Yay.
I didn't know that until you said.
He was born on February 12th, which is today, 1809.
Wow.
And the day is used to highlight his contributions to science and to promote science in general. You can go to darwinday.org to find out more about that. So happy Darwin Day, everybody.
Excellent. Happy Darwin Day. That's right.
In fact, if you go to, he is 200 some years old today.
210, right?
210, right?
Because it's 2019.
Wow.
Yes. Yep. He just keeps getting older and older.
As we all. Yes, as we all.
Well, in this rotation that is Immune, it's Cindy's turn.
It is.
What have you got for us today?
Well, surprise, surprise, we're going to talk about macrophages.
Yay, your favorite. Your favorite.
Yeah, we went back and forth. I was telling Steph that I went looking for papers, and there were just way too many cool things that have come out lately. Yeah. And I sent her a bunch, and she's like, "We have to do all of these."
Of course.
And I was like, "Well, which one are we going to do?" So I picked one called Metabolic Rewiring of Macrophages by CpG potentiates clearance of cancer cells and overcomes tumor-expressed CD47 mediated don't eat me signal. So giggle, right? So
right.
Hopefully everybody's giggling a little bit. I always get snickers when I talk about eat me signals and don't eat me signals and find me signals in my immunology classes, because they're college students, like hee hee hee. Right? So this is a don't eat me signal. So I think this paper was really cool because, one, it's macrophages, which I love. Two, it's CpG DNA, which is something near and dear to my heart that I've been working on for a long time. And I like the idea of macrophages being able to kill cancer cells, and so how cancer cells overcome those mechanisms for macrophages to kill and eat them, what exactly those are, and how can we use those to better understand how cancers are evading immune responses, and how we might overcome those ourselves with therapy to try and treat cancer. So I thought this was rather interesting. So it was published just this year in Nature Immunology. And the authors are, now you're better at this than I am, Vincent. So Mingan Liu, Roddy O'Connor, I can say that one, Sophie Trefili, Kathleen Graham, Nathaniel Snyder, and Gregory Beatty. And so they're from UPenn and Drexel. And I also like this paper a lot because it talks about biochemistry. That just scares me. So I'm going to tell you that right off the bat. So I very am likely to say many things wrong this episode because biochem is, I took it and it's been a long time since I took biochemistry.
The biochemists can email us with their, praise or follies. So, and you automatically get A+ for attempting the biochemistry. But when you're asking me which paper to do, I felt somebody had commented that they really enjoyed when we talked about our own research within the context of the paper. It kind of helps put it together. And I thought, well, since you really like macrophages and this is, an aspect of your lab, and plus the don't eat me signals, it's a good arc talking about cancer cells in the past and inhibitory markers. So I think, it's a good fit.
Yeah, and I will give it my biochemistry attempt. I don't know if I deserve an A+. You can wait until the end to see. But definitely one of the things that has been hot now for a couple of years is looking at how cells metabolize glucose and how that influences their function in immune responses. And it started really looking at T cells, but has become a major player in understanding macrophage function as well. And I guess it's very fitting because Friday we went and did one of these assays for the very first time in some of our experiments. So I kind of sort of understand the assay that they did in part of this. So I'll try and explain it as best I understand it. So just to start off, why are they interested in any of this? Well, so cancer is clearly a big problem. I don't think we need to go that far back, but macrophages are able to kill cancer cells. So they have this capacity, but there are a number of things that tumor cells do to thwart those immune responses. And so we have tumor-associated macrophages that have this potential to kill the tumor cells, but they're suppressed somehow by their microenvironment. And really, that's one of the things that I'm really interested in my lab right now, too. What are those microenvironmental cues? So this was also very interesting to me that way. And so they're capable of this, but they don't do it. And the other thing they can do is they can be reprogrammed. So macrophages are really adaptive cells depending on cues they get. And so tumor cells can secrete and make cues and modify the microenvironment to actually, I guess, sort of like put macrophages in a trance, right? And to bring them over to the dark side and have them promote the cancer growth. So not only are macrophages capable of killing the tumor cells and they don't do it, but then the tumor cells make the macrophages promote their own growth.
Right. So the cancer cells are taking advantage of this massive plasticity that the macrophages have, which is amazing. I mean, I think, when we in the past have learned about macrophages, it's like, well, they go to the tissue and that's what they do. And they phagocytize and they clean up the dead cells, but they really differentiate depending on what tissue.
Exactly.
Yeah.
And they're really abundant. So they're a really great potential target. So if we could redirect them away from the signals that they're getting from the cancer cells and turn them back against the cancer, there really is a capacity to try and eliminate a significant tumor burden in an individual.
How does a macrophage promote tumor growth? Is it by the production of cytokines, say, that stimulate growth?
Yep. So one of the things that they can secrete is tumor growth factor, TGF, and TNF as well. So metabolically creating an inflammatory microenvironment that promotes the tumor growth, as well as specific cytokines and other factors that the macrophages secrete in that microenvironment. So it's almost like the cancer cells are making the macrophages zombies to make their, secrete what they need to live. The cancer cells need to live. Mm-hmm. So there's these macrophages and they have this ability to recognize these cancer cells, and they are certainly capable of eating the cancer cells. But as we know, cancers are successful because of the fact that they have lots of different mechanisms to evade immune responses.
Right.
And so one of the major ways that they do this is they put markers on the surface that tell the macrophages, don't eat me. So there are eat me signals and don't eat me signals. And so macrophages, you mentioned that they clean things up. They clean up the tissues. They clean up dead cells. So they're like janitors sometimes, right? And so how do they recognize those dead cells? Well, so when a cell is dying, it's going to leak ATP. And that is considered a find me signal. So just like a chemokine or a chemotactic cytokine, so just like sniffing a gradient to try and find where the source of something is, ATP will leak out of dying cells. Macrophages will follow that ATP signal to find the dying cell. Now, when a cell stops metabolizing properly, they don't have enough energy to maintain the asynchronous outer membrane protein distribution that they have. And so when that happens, there are what's called flipases that keep phospholipids asymmetrically dispersed on the outer side of the outer membrane and the cytoplasmic side of the outer membrane. And when those flipases aren't propelled by energy anymore, the phosphatidyl inositol or phosphatidylserines will flip. And so they're expressed on the cell surface when they normally are not. And so this is an eat me signal. So when a cell is dying, they're going to secrete the find me signal, and they're going to flip these signals up onto the surface. So let's say the eat me signal. So that's one way in which macrophages recognize cells to kill. Now, in addition to these eat me signals, there are don't eat me signals. And so cancer cells have utilize this mechanism to put this molecule called CD47 on the surface. And so if the macrophage engages that CD47, it tells the macrophage, don't eat me. And so obviously one big thing you could do is block that interaction, right? So the CD47 on the cancer cell will bind something called the signal regulatory protein alpha, or SIRP alpha, on the macrophage. And when that engages, the macrophages don't eat the tumor cells. However, monotherapy, like just putting anti-CD47 antibodies in, as we'll learn, they repeated that in this model in this paper, doesn't work very well. Right. So just blocking that doesn't allow the macrophages to eat the tumor cells more. So the question then is, what else can you do to drive this response? So what can we do to overcome these signals that the cancer cells are giving to the macrophages so the macrophages can then eat them? Well, this comes
is this CD47 tumor specific, or
nope? So a lot
on
of
other cells.
Other cells, so I didn't look that in great detail, but my suspicion is most healthy cells will express this so that they're not getting eaten. For whatever reason they might get targeted, they won't be eaten. I don't know if viruses use this, because that's, viruses and cancers often use a lot of similar mechanisms to try to evade immune responses. And I don't know if viruses upregulate this molecule.
Do you know that with that monotherapy, was that specific to pancreatic cancer? I mean, in this example, it seems like it is, but is that because pancreatic cancer is one of those cancers that has a really high mortality rate? I didn't know if that was one of the reasons why. Maybe there's multiple interactions preventing tumor cells to be phagocytized.
So I'm looking while you're talking, and it looks like it has been used to target a variety of different human cancers and malignancies. I'm not sure all of them, but in this paper they use the pancreatic cancer, but I think there are other ones in which they've targeted it, and probably it does work in some situations. In this particular one, it didn't. So
right.
So one of the ideas that you could kind of exploit is toll-like receptors to promote the activation of the immune response, because these are the receptors that are important. We've talked about them before, that recognize different microbial patterns to induce the immune responses. And so CpG-DNA is one of these. So everybody says, what is CpG-DNA? It's just cytosine, guanine, two nucleotides next to each other. There's a phosphate group in between, which is always there. So why they didn't just call them CG instead of CpG, but that's the nomenclature. And so they were originally identified as a highly overexpressed in bacterial DNA. And so if you chop bacterial DNA up, it was stimulatory for cells. And when people mapped what was the active fragment in DNA on immune cells like macrophages or just peripheral blood cells, they found that you had to have a minimum of a 6 nucleotide sequence that had to contain the CG, and the CG could not be methylated. And that's sort of important because in our genomes, the vast majority of our CGs, the C is methylated, because we have these methylases. And this was a caveat that all along was bugging a lot of people when they first characterized this, and they said, well, this is bacterial specific. Bacterial have these CG sequences and they activate immune cells, and we don't see our own DNA because we don't have these. And the secret was that we do. So those of you who are aware of this, CpG islands are a major mechanism for regulating housekeeping genes for them to be highly active transcriptionally. So we do have CGs, and we do have these 6-mer motifs, although they're much less frequent. But it turns out that, in fact, over the years, we found that, yes, our own host DNA can trigger these too. That was a side story. But the idea here is that you can use stimulation with a CG dinucleotide within the context of a specific sequence to trigger a TLR, and that TLR is TLR9. And this TLR stimulation will induce macrophages to take on a very M1-like phenotype. So this is a type of macrophage functional profile that's generally anti-tumor.
Mm-hmm.
So you think, well, maybe you could block CD47 and activate the macrophage at the same time, and maybe that would be enough. So that's part of what they're going to look at in this paper. And then the third thing that we need to talk about is the biochemistry. So I delayed as long as possible. So now we're going to go into this.
I like on the notes, it's TLRs, yay, biochemistry, yikes.
I did do that on purpose.
I love it.
So the metabolic state, as I said, the metabolic state of cells is a really hot topic right now, and cancer cells specifically. So cancer cells use a specific glycolytic-like profile. They're not very, I think they're not very oxidative. And so, I have that backwards. I might have that backwards. So the idea is if you restrict glucose, so you're not eating a lot of carbs and you're eating more protein, you can starve your cancer. So you could potentially, at least partially in diet, modify cancer cell growth. Although it's not, it's not black and white. I'm not telling everybody out there only eat protein because you're going to starve of cancer. You'll never get cancer. That's not true. But the idea is in cells that require a really high metabolic rate and a lot of production of amino acids, lipids, and nucleic acids, they need to have a different type of metabolism than in cells that are just, surviving, because they need rapid energy sources to produce a lot of ATP. And then they need this shunt to make all these other structural molecules in order to sustain enhanced proliferation. So cancer cells are some of those. And if you can imagine, T cells are another one, because T cells can proliferate from one cell to thousands of cells over the course of a few days. Right. It can proliferate faster than in a 24-hour period. So that's another thing. So here's your primer. If you remember anything from your biochemistry, there's oxidative phosphorylation, and that produces a lot of ATP using glucose as a source for acyl-CoA. But it takes a longer, but glycolysis will rapidly produce ATP. But the cost is you produce a little less, but you can produce it rapidly. But you can also shunt some of the intermediates off to make all these other metabolic things that you need, like nucleic acids, lipids, and amino acids. So when a cell needs an immediate form of energy and rapid proliferation, high metabolic rate, they tend to switch to this glycolytic pathway. But they still need a source of this acyl-CoA, because in oxidative phosphorylation, glucose is broken down into pyruvate, and that makes acyl-CoA. And so that's how you get the shunting into the TCA cycle. But to get to the TCA cycle the other way, without using the glucose as the main way to get to pyruvate, you can break down fatty acids. And so it's fatty acid oxidation. And so when you use fatty acid oxidation, you can shunt to this pentose phosphate pathway. You can produce more reactive oxygen, reactive nitrogen. So it's metabolically a little bit more destructive that way. But there's, this idea that you can get more energy and more metabolic intermediates that you need using this way.
Right.
This is also a big topic, a growing topic in virology, because we now understand that, when a virus infects a cell, a lot of things have to be made, right?
Oh, yeah.
A lot of parts. So you have to crank up the thermostat. And so we're starting to learn how, viruses manipulate, how they can push metabolic pathways in certain ways. So they get a lot of nucleotides, or if they have an envelope, they have to make fatty acids and lipids and so forth. And there's some really amazing strategies that are being uncovered. People are getting back into metabolism and virology. And in fact, last year I added a new lecture to my virology course where we talk about metabolic changes in infected cells. It's really great.
Oh, that's cool.
It's if you paid attention in biochem, now it's paying off.
Yes. Because I know that's a very common thread amongst undergraduates, or maybe even high school students, when they learn about the Krebs cycle or oxidative phosphorylation. They're like, when am I going to need to know this? Well, turns out, if you're listening to this podcast, but also, I think that you guys are right. That this is combining these type of things with single cell technology. We can learn about what individual cells are doing. And what we'll learn about in this paper is a lot of the subsets of the cells and immunology that we defined, let's say 30, 40, 50 years ago, aren't necessarily holding up over time. And there's a lot more nuance. And I think, just looking at surface receptors anymore, we know isn't going to help define these subsets. So looking at things like metabolic profiles, maybe that will help us kind of divide up subsets more and be more fine-tuned with our therapies.
Right. I welcome that. Get away from the cell surface.
Yeah. Right.
Yeah.
Too redundant.
So I put a picture in here to confuse Vincent and Steph.
Nice picture.
Maybe you'll know it.
But yeah,
so the end result is that really how you use glucose once it gets into the cell, and where the acetyl-CoA is coming from, and how that's being used in the TCA cycle, and then what the product's coming out the other side. So if those individuals who paid attention in class in biochemistry and followed that over time instead of getting off on all these other things that we've studied, they thought, well, what if you could measure these things? And they came up with the most simple thing, right? So they measure either oxygen consumption or pH change. And that will tell you whether or not you're using oxygen to fuel the ATP production complex on the mitochondria, or whether you're doing fatty acid oxidation and producing acid and lactate out the other side.
Right.
Mm-hmm. And that's it. And there's this couple $100,000 machine that does this.
Of course,
and everybody's buying them.
Right.
And we're all doing these assays.
So you have one of these in your lab, you were saying? Or
I don't have
in
it
your
in
core?
my lab, but we have a core. So we got one a few years ago, and it's actually gotten old now. And so we just got...
Isn't that crazy that that's 2 years ago and now it's old?
Yeah. And so now we got two brand new machines because this metabolic core is supposed to be a big deal. So they're putting a lot of money into it at Cornell and actually giving us instrumentation. So we have a 96 well and an 8 well. And the way this works is you seed the cells in this special, chamber that you have to spend a fortune for so that you can plate them in the thing that goes in the instrument. And you just plate the cells in there, and then you have to change them to a media with no buffer because you can't buffer your pH change. These are detecting the minutest little teeny tiny change in pH. So we have to make sure that everything is perfectly set for this. And so you put the cells in this thing, and you have to equilibrate this little sensor plate. And then you put the sensor plate on the cells, and you put it in the machine, and then it just runs everything. And it automatically injects in drugs at specific times, and then measures the oxygen consumption and the pH change over a few minutes following the injection of a drug. And it's the clever part of it is, which drugs do you use and when do you use them? And when you inject things in, for example, you guys can follow along a little bit here. So if you inject in something called oligomycin, what that does is it blocks complex 5. And so the oxygen can still be used, but you don't get ATP out the other side. And so you can look at you can read the basal respiration rate first, and then you add oligomycin, and you can look when the change in the oxygen consumption will tell you the ATP production rate. And then you can treat the cells with something called FCCP, which basically short circuits the entire ATP production complex. And so it produces the maximum amount of oxygen consumption rate and acidification, because the protons are just going back and forth, and there's no energy actually produced. And that'll tell you the maximal respiration rate. And then you can add 2 drugs called rotenone and antimycin, and they'll block the early steps in the ATP production complex. And that just basically drops all of the oxygen production rate down to background. And so anything left after that is respiration that's not due to the mitochondria complex there,
Right.
the oxygen production, ATP production complex. And so you can look at over time after adding these different drugs, you can see the basal rate and then the rate after ATP production and then the rate after ACCP and then the rate after rotenone. And so then you can look and measure all of these different things: maximal respiration, ATP production, basal respiration, spare capacity, all these things. And they tell you something about how the cell is using its metabolic process. Now, this is just one. This is called the mito stress test, and they have lots of different ones. And so this one is, probing the different parts of the mitochondrial membrane complex. But then you can also add glucose and modifications of glucose that block glucose metabolism, and you can read things out from there as well. And so this is oxygen consumption rate, but then it also measures pH change. So you can also look at the what's called extracellular acidification rate, or ECAR. And so you can look at the traces that come from this OCR and ECAR, and that will give you an indication of whether the cells are using fatty acid oxidation or oxidative phosphorylation. And so it's actually quite a powerful system. And there's a lot of different kits you can buy that can probe all the different aspects of cellular metabolism. This is just looking at ATP production and oxygen usage. But generally they're using these kinds of things to say, okay, well, cancer cells use a lot of this one and macrophages use a lot of this one. But then if you trigger macrophages, they change their metabolic profile, and T cells are one profile when they're resting and then another profile when they're proliferating. And so all of that together is really interesting. So it's a lot of background, but we had eat me and don't eat me signals. We had ways to stimulate cells like CpG-DNA and other TLRs. We had a little bit of a primer on the biochemistry and how you use glucose inside of cells and how you measure how a cell is using glucose. So what do they actually do in this paper? So first they were using a mouse model of pancreatic cancer, as Steph mentioned, and it's syngeneic, which means it's from the same, strain of animal. And in this case, they're using mice, and immunocompetent. So they're not using, a lot of times when you do a cancer model, you have to use a nude mouse or a B cell deficient or T cell deficient mouse. And in this case, it's an immunocompetent mouse. And so these tumors grow in the mice, and if they add anti-CD47 as an immunotherapeutic, there was no effect on a tumor. And so, so it could be due to the cells not phagocytosing, macrophages not phagocytosing, but it doesn't seem to be the case. The antibodies didn't really do much in vitro either. So they just said, okay, well, let's start playing with this. Let's see if we can do a combination therapy. Let's add a TLR ligand and the CD47 and see if they could do anything. So if they stimulated with TLRs, none of the TLRs enhanced the capacity of macrophages to eat the tumor cells in vitro when you add anti-CD47 antibody, except CpG-DNA. So then they put the tumors implanted them in a mouse and waited 6 to 10 days. And that's important because now you have an established tumor. Right? Because a lot of immunology papers, when they're looking at cancer papers, they start treatment right when you put the tumor in. So it's basically looking at, can you prevent the tumor? It's much more difficult to get something to work if you allow a tumor to get established and then try and treat it, which is more biologically relevant to a human. So they allow this tumor to establish, and then they add the CpG-DNA, and lo and behold, the CpG-DNA reduced the tumor volume. They showed nearly flat growth curves for a while. They enhanced survival, but eventually there was relapse. But CpG showed promise, right?
Right.
So how did it do it? Why is CpG-DNA doing this? So it seems to reduce tumor growth. And so it increased inflammatory cytokines. They could measure this in the sera of the mice. And there was an increase in the number of tumor-associated macrophages, which would suggest that they're getting there. And there's an increase in the macrophages that have tumor cells inside of them when they look in the tumors. So somehow when you treat the mice, and they were very specific about saying they were treating these mice IV and not IM or sub-Q. A lot of people do that. And so they think that this had some better mechanism of working. I don't know really how that would work.
It could maximize like the delivery because it could
probably
reach the tumor better. I would
Yeah.
assume.
So, I think that the vascularized tumor also is going to, the CPG is going to get into the tumor and it could act on those local macrophages, probably part of what it's doing.
Right.
So they see these, so the CPG has an effect. They see the macrophages increase. They see the macrophages eat more tumor cells. But, what you just asked was, where is this being delivered? So where is the CPG actually acting? So they asked, is it actually the tumors associated macrophages? Or could it be all the other macrophages in the body that are doing something? So they used a drug called GW2580. It's an inhibitor of CSF1 receptor. And so that would prevent any new macrophages from getting into the tumor. And so when they did that, it didn't have any effect. So they think that the macrophages that are already in the tumor, once the tumor establishes, the ones that are really critical for when you add CPG, they're having this effect.
Well, that would make sense based, Cindy, on what you said in the past, that when you have these tumor-associated macrophages surrounding the tumor, they're almost like in a trance. And so they are specifically being affected by not the peripheral macrophages. So that does make sense.
That's right.
And all these Macs have TLR9, is that right?
In mouse, they do.
Well, and it's interesting because they don't really ever look at TLR9. When you do a search of the word TLR9, it's only in this paper 3 times, 2 in references, and 1 to describe CPG. So I think that's something that we will talk about later. But yeah, they don't pay any attention to TLR9 in this paper. No TLR knockout mice, no TLR knockout macrophages, unless I missed it in the supplemental.
No.
Okay.
No.
But that's maybe for later. We can work our way through the paper.
Yeah. So there's 2 things there. We can address it now. Might as well.
Sure.
One is that I had written something when I was going to talk the intro and I jumped over it, and that is there are other DNA sensors, right?
Right.
And Vincent, you know this really well. So they're cytosolic sensors for DNA that see DNA viruses, right? Sting, C-gas, and things. Yep. But, it's less likely that if you inject CPG into a mouse that it's going to make it to a macrophage, and that macrophage is going to take up that CPG and then somehow shuttle it into the cytosol. So it's probably more likely to be TLR9 dependent, although they did never test that, so they can't say that for sure.
I don't know. It seems to me, I mean, if you don't want to pay the money for the TLR9 knockout mice, just get a cell line and just try. I don't know. That probably opened up a can of worms. I'm assuming maybe they did try, and there are probably different mechanisms. I don't know. It just seems suspicious that it was missing.
Yeah. And the other thing that we have to put out there that I was going to save for the very end, but that I kind of like when I answered your question the first time, I sounded a little funny. And that's because human macrophages don't express TLR9. So this is a beautiful study, but how relevant is that going to be for humans? I don't know. So if CPG is the only one that works out of the TLRs they tested, and there's no TLR9 to see CPG in a human, I don't think this mechanism is going to work. Does it negate the whole study? Absolutely not. I think that the bigger idea here is that the modulating the metabolism of a macrophage to thwart the negative impact of a cancer cell on the macrophage's ability to kill it is the important part.
Definitely. And the fact that we don't know if it's TLR9 still leaves the potential for an intracellular signaling sensor like, Sting or C-gas. So I think definitely still relevant in terms of metabolic states of cells. But yeah, that is interesting. So in humans then, TLR9, it's on B cells and DCs or
it's not just any DC. It's a one unique special kind of DC called a plasmacytoid dendritic cell. Yeah,
gotcha.
Yep. So it might be very low expression on a few other cells. Somebody probably is working on that, listening to this and saying, "My cell expresses it." but the 2 major expressors are B cells and plasmacytoid dendritic cells, right? So where were we? So we said they've narrowed it down to the macrophages that are actually in the tumors that are responsible for mediating this CPG-induced protection. And then they also looked at PD-1, which we've talked about immune 14 for immunotherapies, and it didn't seem to have any effect on that because CpG is known to induce, I guess in some cells it can induce expression of PD-1, but it was not changing the expression of PD-1, inducing or reducing expression of PD-1 or PD-L1 in the cancer cell or the macrophage in the tumor. So it wasn't that. And they also went one step further and used what are called RAG2 deficient mice just to show that it's not T cells and B cells. This really is an innate cell. All of these data together said that this really was macrophage. We have this CpG treatment via macrophages is protecting the cells, and it is allowing the macrophages to kill the cancer cells. And so they looked at bypassing CD47 signal introduced by the tumor cell. So they tested a bunch of different tumors that both had and didn't have CD47. And what they discovered was this CpG-induced protective effect was totally independent of CD47 expression. So that might explain some of the initial observations where if they just treated with anti-CD47 antibody as an immunotherapy, it wasn't working in this model.
Right.
Because when you add the CpG, it doesn't matter. This is all CD47 independent. So don't eat me doesn't matter. I don't know how we fit that in there. So if it's not dependent on these eat me or don't eat me signals, but CpG is enhancing the ability of cancer cells to be eaten by macrophages, what exactly is going on? So I don't know if they tested a whole bunch of other things or they just said we have a seahorse analyzer and we'll do some studies. But bottom line was they started looking at macrophage metabolism. And they argue, there's a lot of people looking at macrophage metabolism, and M1, M2 have different metabolic profiles, and so this might be important, which is all true. But the bottom line is, so they stimulated cells with CpG DNA. And if you stimulate cells typically with a TLR ligand, we think of that as a very strong M1 inducer. And everybody said for a long time, M1, M2, this dichotomy, this strict either or designation is kind of not really realistic because there's a spectrum of macrophage phenotypes, as you mentioned, Steph. Right. And so when they stimulated with CpG, they were a little bit more careful than some other people to look at exactly what were those macrophages making. And it turns out that they made some things that were made by M1 cells and some things that were made by M2 cells. And so they didn't really look like M1 or M2. And some of the markers that you would typically associate with one or the other, they didn't change expression at all. So they had this really weird in-between phenotype. And so then they said, okay, well, if you stimulate cells with LPS and interferon gamma and they become very strongly M1 and this causes them to increase their oxidative phosphorylation or whatever, and you stimulate them with M2, they go the other way, or I think it's vice versa. Sorry. It's confusing. So what is CpG doing? Because we always thought of that as an M1 marker, an M1 stimulus. And so what they saw was that all the TLR ligands increased this glycolytic flux or this ECAR, so the pH increase. And they use this, seahorse to measure this and everything. But the interesting thing was, and the sort of unexpected thing was that CpG, but not the other TLRs, also increased the basal oxidative respiration, reduce the spare respiratory capacity. So it's doing both. It's changing the metabolic activity both directions, where we typically think it goes either this way or that way. It's doing both. So it again now has this unusual kind of intermediate phenotype. So it's sort of in between an M1, M2, and it's in between glycolysis and oxidative phosphorylation.
Can we call it M1.5?
I don't know. That sounds good to me. Or X, I don't know. I think there's already one of those, but yeah. So it's this in between thing, and it's got markers of both, and so that's interesting. And since CpG was the only one that had this enhancing effect on using anti-CD47 antibody in this pancreatic cancer model, does this actually have anything to do with anything? And so they hypothesized that maybe this unique metabolic pathway activation was important for the anti-tumor activity. And so they start probing this by looking, they first see if there's more mitochondria inside the cells that are stimulated with CPG, which I was going to do. I've been looking forward to doing this experiment. They already did it.
Oh, no.
But that's okay. Because, this is these beautiful dyes that you can add that will stain the mitochondria, and you can image them in live cells, and you can look at them fizz and fuse, and you can count them, and you can see how big they are and everything. And so I was all excited to do this, but they've done it. Anyway, so the bottom line is that they have more mitochondria, which fits with an increased oxidative phosphorylation rate, OCR increase, right?
Right.
And then they started probing fatty acid oxidation, so it's the other way. So they treat with a drug called etamoxir. Do you guys know how to say that?
I think you said
it. Yeah.
I think, that's as good as I would have said it.
All right. So it inhibits a protein called carnitine palmitoyltransferase 1A. Anyway, when they did that, they blocked the ability of the CPG to change the OCR rate. And they also reduced the ability of CPG to enhance phagocytosis of tumor cells in vitro. So I thought, that's kind of cool. The fatty acid oxidation pathway seems to be connected to the phagocytosis of tumor cells. So what would you do next?
You would give exogenous fatty acids to see what happens.
I guess you could do that. They did something different. They gave the atoxamir in vivo and see if it affected the tumor. And it did. So if they blocked with atoxamir, they blocked the ability of the CPG to protect against the tumor, even though there were the same number of macrophages in the tumor. So the CPG somehow, its ability to change the metabolic respiration of the macrophages affects its ability to phagocytose the tumor cells, even in vivo. So altogether, this shows that this fatty acid oxidation induced by CPG is critical for the anti-tumor effects by increasing phagocytosis of tumor cells, and that all of this is independent of this eat me signal via CD47. So.
There was something I was curious about in this section before you had gotten to the looking at fatty acid oxidation. They did say that the CPG they did upregulate PD-L1 on the macrophages. And so while we usually think of PD-L1 on the tumors, do we know much about macrophage T-cell interactions? That if you're upregulating PD-L1 on macrophages?
If you upregulate PD-L1 on macrophages, I would think you would be suppressing some T-cell responses.
Yeah, that's what I was thinking, that maybe the unintended... I mean, we'll see they did take out the T-cells and nothing changed, so I guess that tells you that it's fine. But it's interesting that it was upregulated, which we think of as another inhibitory signal, so maybe some potential for... I mean, it seems to be fine, but I thought that was kind of interesting that the tumor didn't change, but the macrophage did.
That's true. I forgot about that point. Yeah.
I mean, it says data not shown. That's why. So I don't. Yeah. Maybe that was on purpose. I don't know.
I don't know. Yeah, I tend to ignore that when they say data not shown until I see it.
Right, exactly.
So it might be really a small amount change, but so in general, macrophages in tumors are immunosuppressive, right? So maybe it's affecting some aspects of the the suppressive activity of macrophages, not other aspects.
Right.
That's true.
So when you do fatty acid oxidation, what product of that is important for the CPG effect? Is it just getting energy? Because that's a way to get energy, right?
It is a way to get energy. That's the part they leave out. And I don't know if you guys read the discussion. I read it and I didn't see where they address that at all.
Yeah, no, they don't really. I mean, fatty acid oxidation will give you more acetyl-CoA. So, that can help the TCA cycle.
Well, they suggested that might go to cholesterol biosynthesis, which
and the
you need
fluidity
for
of the membrane.
Right, because that very last experiment. Mm-hmm.
That's right. I forgot about that.
They did the Brofeldin experiment.
Right. Yeah.
Tell us about that.
Yeah, they blocked the ability, excuse me, of I can't remember which chemical
Brofeldin A.
Yeah, but thank you, Brofeldin A.
So that blocks secretion from the ER.
Right.
Yes.
And so when you treat cells with profeldin A, it inhibits the CpG effect, basically.
Yes, yep.
Because they don't phagocytose as well, because you need membrane fluidity for that. So maybe, maybe the fatty acid oxidation and getting cholesterol is related to that as well.
Mm-hmm. Right.
That's true. Although we have also looked at intracellular trafficking of TLR9, and when we treated with profeldin A, TLR9 didn't traffic to the right compartment in order to be able to see CpG DNA. So it also may be a ligand access issue. So it may be true, and not for the right reason. But
it's true. Yep.
Yeah. So that's why I got confused, because I blocked that out because I was like, I don't know if I believe that. But so regardless of whether it's membrane fluidity or intracellular trafficking, clearly if you block that pathway, it's interfering with the ability of the CpG to have its effect.
That's a great point. I think they kind of think it's fluidity, but you're right. It couldn't just be getting the TLR to the right place. Yeah.
Right.
Makes sense. Yep.
So the last thing they did in this paper is they really went into a deeper dive into where the fatty acid that you use to increase the fatty acid oxidation, where you're getting the acetyl-CoA from following CpG stimulation. And they showed it was this lipogenesis and not free fatty acid uptake. So there are two ways to get more fatty acids to metabolize to get acetyl-CoA for the fatty acid oxidation pathway. So you can take them up from outside of the cell, or you can make them or degrade lipids that you already have and chew them up to make it. And so it's not taking it up from outside, it's intracellular sources, which is interesting because if I understood what they were saying correctly, the uptake from extracellular sources is a typical way that macrophages get lipids to support fatty acid oxidation. So one of the things we've done is looked at different uptake of different fatty acids by cells. And when you differentiate them or treat them in different ways, they definitely have a different capacity to take up lipid. And so M2s take up a lot more lipid than M1 cells. And we tend to call them not M2 cells, we call them foam cells, which is a different name, but it's related to atherosclerosis. So macrophages can have different phenotypes in atherosclerotic plaques as well when they take up a lot of exogenous extracellular lipid, and they can use it for this metabolic pathway as well. But it seems like in this case, for whatever CpG is doing, is ramping up the metabolic pathway to use the intracellular stores of lipid to shunt into this fatty acid oxidation to generate energy.
So that might argue against a ketogenic diet, correct? Because if you're it wouldn't really matter if you're adding fatty acids.
No, it wouldn't matter. So you can starve yourself and do that. But at least for your macrophages, it might not make a difference. I believe, and I can be corrected, but I believe for cancer, it's the other way. They do take up lipids from outside. And then that shunts into this pathway and provides energy for the cancer cells.
Right.
Yeah.
So they note in the discussion that in people, CpG has had limited efficacy, right? And they argue that it's because it wasn't given systemically like they did in this experiment.
I don't believe that. But yeah.
But you would say because there's no TLR9 in people, right?
Yes, that's right.
Right.
But is there another TLR that would recognize CpG?
No. There's not another one that would recognize CpG, but 7 and 9 are very similar. They both strongly induce type 1 interferons. I don't know, but could test whether they induce the same metabolic pathways. Now, I have to go back and look. Did they test 7? I think they tested 2, 4, 3.
Yeah, they did.
They did 9. They did test 7 and it didn't show up.
Right.
Yeah, here it is. For the gelatin, they tested imiquimod, but they wrote it L-miquimod. They typed it wrong. So
2, 3, 4, 5, and 7.
Yeah. So imiquimod, that's only one of many ligands for TLR7 that in theory should all work the same way, but I don't know if they do all work the same way. So 7 is potentially still a viable alternative. But there are other ones that are much less well studied, 11, 12, and so forth. So the jury's still out.
So what's next with this, Cindy?
So what do you do? Well, this was really a complicated paper. And once I started reading it, I was like, what did I get into? But I think, the idea that, metabolic status of cancer cells and immune cells is really important. And we don't really fully understand what's going on, but some, probing or inhibiting different parts of these metabolic states could alter macrophages within a tumor. And at the same time, you could have similar or opposite effects on cancer cells that if you're altering the metabolism of both of these, maybe, just maybe, it could have some therapeutic benefit. Would it ever work on its own? Probably not as a monotherapy. But if you can, slow down cancer growth and ramp up the immune cells, maybe that's sort of how we work. Should that also be combined with something like PD-1 and other checkpoint inhibitors?
Mm-hmm.
Maybe this is combining it with CD47, but maybe you get a different outcome if you combine it with PD-1 blockade. When we said CPG was inducing PD-L1 on macrophages. Maybe if you block that interaction on T cells, maybe if you can have the T cells active and the macrophages active, you even have something better.
Right. Or maybe it's not the CPG focus, but just the fact that they discovered this intermediate macrophage phenotype that, seems to, when it's stimulated, kill tumor cells. So is there another stimulant that can upregulate? I mean, maybe that's the way to go since TLR9 isn't on them in humans.
I think that's exactly right. You could find a drug that might have a similar effect on the metabolism, maybe. Yep. Right now that you sort of know what to look for.
Yeah.
Seems to
but it would have, remember, if these things are not going to be specific too, right? So they are going to affect metabolism in every cell, and what effect that's going to have, I don't know.
Yeah, like giving they said they hadn't done intravenous. Is that what you had said before? So, I mean, giving an intravenous CPG, what's that going to do to your B cells and dendritic cells? I imagine there's going to be side effects.
Yeah, there's going to be cytokine storm and things that go with it too.
Right.
They all have their caveats, right?
Right. But it is a very cool paper. I'm glad we went through it. I learned a little bit more about seahorse. I had heard of it, but don't work with it. So that was nice.
Yeah, that was great. I really enjoyed that.
Yeah.
It's quite a lot of work, but
it is really a lot of work.
I think it's presented pretty clearly that you can really get the major points out of it. It's good.
Yeah,
definitely.
And I didn't go through each figure. I mean, we could have done that, but I think generally the points that we discussed were the major points.
Totally.
Because they did a lot of work in this paper.
Right.
Yep,
right.
Absolutely.
Yeah.
All right. Let's see now. We have.
Didn't we give away a book?
Yeah. So last time we said we're giving away a book, and I forgot what the book is.
Something with monocytes.
It's monocytes. People seem very excited about monocytes. And Cindy would say, of course, why not?
Something about myeloid cells.
Yeah.
Oh, myeloid.
Anyway, so a lot of people wrote in. Let's see if we can.
I was so glad to see so many responses. You never know. I mean, you have the numbers, but we don't know how many people listen. So it's nice to see.
Yeah. I mean, for the other podcasts, we get pretty good responses when we give away books. So this is the first one. So for immune. And so it was a great response. So let's see. We have Dahlia wrote in. Chris wrote in. Chris is a PhD student at Indiana University, where we went last year to do a TWIM. And he I've been listening to Twix, and that is probably what pushed me to go to graduate school.
Oh my gosh.
That's great.
I studied
cool.
mass spec under the direction of David Klemmer and share these podcasts with him. He shares my admiration for this sort of scientific media. Thanks for creating a dialogue and explaining the fascinating world of immunology.
Thank you.
Yeah, thank you.
So Amanda is a grad student in Saskatchewan.
Yeah.
And would like the book. I've always been intrigued by the immune system. Immune was my first immune podcast. Still going through the backlog of episodes. Keep up the good work. Anthony, this is a good one. I found the subject of immunology to be an uphill battle. Jeepers, I thought. The only thing I know so far is that B stands for bone and T for thymus, and now I find out that I even had that half wrong. That's really funny.
I know. I love it.
Mitch is also a longtime listener. Sam is a PhD student at NIH and Cambridge University, working on myeloid cells. Trudy, is a listener to all the other podcasts and someone who writes in a lot.
And she wrote, I need this book.
I would be remiss if I didn't take this opportunity to thank all of you for your continued efforts. I'm a patent agent, and the material discussed here is often so relevant to my work that I've listened to several of the episodes multiple times, in particular episodes 4, 7, and 14 about CAR T cells, the complement system, and checkpoint inhibitor therapy.
Oh, that's great. I'm glad we can be of use.
Aditya. Right.
This is a really funny
immunophobes. He's an Australian high school student.
Yeah,
Wow.
This one was nice.
I don't have knowledge of immunology, though I enjoy keeping a sense of smug pretension listening to your conversations as if I'm one of those readers who send in corrections, but I'm not. Instead, your thoughts and ideas travel to the periphery of my cerebral horizons at this Best Western place. Anyway, it's a very funny email.
It's very unique and creative.
And after he wrote it, he said, or she, I don't know.
I totally regret
writing that email.
They said I thought I was being creative, and they were creative, and it was wonderful.
It was
very nice.
Don't regret
entertaining. We love these.
It's a very good. Steph, why don't you take a couple and...
Yeah, sure. Hi, Immunesters. It's -3 here at Thiel College in Northwest PA. Tons of snow. As an immunoparasitologist myself, I'm biased to TWIP and Immune. I make TWIP part of my parasitology course, and will start making Immune part of my course, my immunology course, next time I teach it. And third, I'd love the book.
Hey, I want to know, is this Delbert that I know? Because how many Delberts are there?
That's true.
Yeah.
He was a grad student here, and I wanted to know if that was him.
Oh, right in, Delbert.
Yeah, let me know.
Kevin, I just found this show after listening to the Twivo. It's been great learning about immunology through the show. Cindy, you can take the next one. Short and sweet.
Candace writes, did I win? So Candace is down the hall. So she's also, I'm going to give her a shout out. She's probably going to be very embarrassed and turn red if she hears this, but she's starting her own podcast, and I'm sort of her little mentor for her podcast.
Oh, that's great.
And it's called Excelsior, and it's fantastic. And she's doing an awesome job. And I hope I got it right. And so she's talked to me a lot about immunology. She's incredibly passionate. She's passionate about science communication. And yeah, she's doing this podcast. So
that's great.
Good luck with it. Yeah.
Amy writes, want to thank you for your efforts, science communication, like to enter for the drawing. Immunity runs in my family, so I'm very interested in all things immune. She's been listening to the micro podcast and motivated me to go back to graduate school. I did an undergrad in microbiology. That's great. Sue Ellen, I hope I get the book. Episode 16 was great, but all the immune episodes are great. Let's see. Eric, I'd love to be entered to win a chance. Michael, this is one of my favorite Twix podcasts. There's a couple more. Cindy, you can do some.
Sure. Well, you gave me the long ones.
Oh, sorry.
No. So Matt writes, "Hi, Immunonauts. I always love these. You guys come up with the most creative names. Is this something that's like for all of the podcasts that you do, Vincent? I love it. I love reading these. This is so cute.
I'm going to put a little note to myself. I'm going to go through all 17 episodes. I'm going to read all the names. I'm going to compile them in a separate sheet, and then we can have a list.
What a great idea.
Yeah, I'll do that.
Yeah. Okay. So anyway, Matt writes, I would like to throw my hat in the ring for the contest if it isn't over already. I guess because we only come out once a month, they think it might be over. But so Matt's a technician in the HIV lab, and it would be past time to learn about macrophages and their kin, and this book would be great help. And he also thanks us for the podcast and for this week podcasts in general. They've been a big help as I have moved from more general microbiology to virology, and you can't really learn about viruses without learning about the immune system as well. Agreed. So Sophia writes, hello, immunodecipherers. And greetings from Greece. Greece, we're worldwide. Yay!
That's great.
I love this. It says, first, I'd like to say that the female hosts in the show have such beautiful and warm voices and make it so exciting to learn about immunology. Thank you.
Thanks, Sophia. That's nice.
Honestly, I've always avoided immunology because it's so complicated and I never get what happens in the end, but you guys have made me feel better about this subject and you have so much excitement talking about it that I get hooked. So down to business. Congratulations to Steph.
Woo.
She graduated not that long ago and moved. And I love this. This is so cute. Also, if I win this book, very small chance, I would like to donate it to Cindy since she wanted to read it. And then after she reads it, she can tell us all about it and I will understand more this way.
Oh, that's so cool.
Oh, and so I'll just read the end of this. Finally, another listener was looking for books for lay people that explain immunology. Yeah, we talked about that in one of our one of the ones we did. Okay, so there is quite an old book out there which might be considered outdated or classic, depends on how you see it, but I love the author and would like to recommend it. It is called The Thorn in the Starfish by Robert Desowitz. So wish you a great year of podcasts. Please keep it simple. If you wouldn't mind. I wouldn't mind if you didn't do any papers and just talk about a subject and ask each other questions as you do. Just a suggestion. Thanks again for doing this. And she is 6 degrees in Greece. And so that's the end of our entries. So if you didn't write in, you should have because you had a decent chance.
So we had 17, I think.
17,
I counted
not
them.
bad.
So let me pick a random number between 1 and 17. And the number is number 8.
8.
And I didn't label them. Well, let's see. 1, 2, 3, 4, 5, 6, 7, 8 is the high school student in Australia.
Oh, so that's great. That might take a little longer to get there.
It sure will. So, Aditya, you have to send me your address.
Yay.
You have to email it to immune@microbe.tv. And I need a phone number because international shipping requires that. So even though you wrote a corny email, you won. But that's a random thing. Anyway, we'll have more books to give away in the future. Keep listening so that you'll hear. You have to listen, otherwise you'll never know.
Right.
All right. Let's end up with some picks of the week. Steph, do you have a pick?
I do. So I'll paste it in here. It's called The Pregnant Scholar. And what this is an organization that basically tries to improve the knowledge about women who are pregnant or nursing or parents, but mostly focus on women in STEM, about their rights, particularly their Title IX rights, and really trying to educate broadly administration down to the student level. And it was originally funded by the National Science Foundation, so it was a part of NSF. But there's a team of 2 professors and a lawyer who's the staff attorney. And with the Pregnant Scholar, I am working with the person who's the attorney, and she's kind of like the liaison for on the ground. She's really involved. And with her, we've started this community ambassador program where you can become an ambassador through the Pregnant Scholar to kind of be a point person at your campus for somebody who might be experiencing discrimination if they are pregnant or were pregnant or are nursing. There's a lot of things within the umbrella of motherhood in academia. And this spans from undergraduate, graduate, postdoc. So this is really all encompassing. And so we're really trying to make this something where there's a point person on every campus if there's issues. Because a lot of this is just education and understanding what your rights are and where to go. And so I just want to put that as my pick. And if you know anyone who needs information or support, to get a hold of me, and I'd be happy to help.
That's great.
Could you put the link in there?
Yes.
Steph? That's great.
Yes.
Cool. Cindy, what do you have?
So I was looking around to see which of these many articles I could choose, but I chose one article. Basically, there's an increase of over 500% for immunization against measles in this county in Washington State where they're having an outbreak of measles. And so it's really, the measles outbreak is getting so bad that the anti-vaxxers are vaccinating. I made that title. I should have written an opinion article on titled that because I think that's great. But I think what it calls to light though is that, it's easy to say that we don't want to vaccinate, when things are great. But then when things are not so great and we're having outbreaks, people are realizing how important this is. And so there's an increased demand, and it's sad news, but great news that these people are getting immunized. And I debated between this too. There are, because these outbreaks and these students are hearing about this, there are kids that are trying, teenagers, I would say, that are trying to find out ways to get immunized without their parent consent. Because if they're under 18 or whatever, and they're still considered a child, they need parent consent. And the parents are not immunizing them because it's against their beliefs, and the children are begging for the vaccines. So it's an interesting state in which we are in right now, as far as the landscape of disease outbreaks and things that can be completely prevented by vaccines, and people's attitudes towards vaccines, and perhaps how they're changing, and that might be good.
Yeah.
It's just amazing that the anti-vaxxers started this outbreak.
Yep.
And now they're getting vaccinated, which is great, but you could have prevented the whole thing.
Yep.
Right.
By just vaccinating.
Yep.
Nothing wrong with vaccinating. It is so bad. It's just amazing.
Yep.
My pick is a website, a nonprofit organization called the Center for Science in the Public Interest. And this was founded in 1971. It's a science-based consumer advocacy organization which is focused on improving the food system to support healthy eating. So they provide advice which is science-based.
I love this idea.
Great.
I came across this today in a New York Times article. They mentioned it about food. So it's really good. They have a great agenda. They're nonprofit. They do a lot of important things. They have articles like, what you should eat, what you shouldn't eat. Coconut oil, probably ketogenic diet you can find here as well. So Center for Science in the Public Interest.
Yeah, and they have a tab there, Eating Healthy, and it's science-based information about ingredients, foodborne illnesses, foods to avoid, what to eat. I'm going to read this for sure.
Definitely. And within the, because a lot of times I always go straight to the biotechnology section to see what their views are on GMOs. And what I really like about this is they just address head on, hey, here is the science. Here's why they are different. But here's why they are safe. And I like that they're kind of upfront with that, but yet they state, this is our position. They're safe.
Yeah.
Great.
All right,
Thanks.
That's Immune number 17. You can find it at microbe.tv/immune or on any podcast player that you use to listen to podcasts. Please subscribe so you get every episode, and we know how many people are listening. That really helps us a lot. Send your questions and comments to immune@microbe.tv, and keep listening. We'll have another book contest soon. And if you really like what we do, consider supporting us financially over at microbe.tv/contribute. Immune is Cindy Leifer from Cornell University. You can find her on Twitter at Cindy Leifer. Thanks, Cindy.
Thank you. See you next month.
Steph Langel is at Duke University, and on Twitter, Stephanie Langel. Thanks, Steph.
Yeah, thank you. This is great.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.